Welcome to our video series for hands-on tour of the single-cell analysis journey. My name is Juan. In this video, we'll learn how to assess overall quality of our single-cell gene expression assay using web summary file output from CellRanger. Before diving into the web summary, let's first go over how can we find the web summary file from the cloud analysis. In the earlier session of this video series, we have completed the tutorial on how to use 10x cloud analysis and analyzed the neutrophil dataset. We analyzed this dataset in two ways. We analyze the dataset with default parameters, and also we analyze the dataset using custom parameter, which is by using the force cell option. We will start assessing the results using the custom analysis result, and then carry out some comparisons at the end of this video. After the analysis completes successfully, you can click into the analysis to explore the results. There are two ways to access the web summary file. We can click this button to see the results summary. We could also check the summary HTML file and then download in browser. Then we can view it in any browser of our choice. So now I'm just going to click the See Results Summary to open the web summary file. On the top of the web summary, we can first confirm the pipeline used to generate the results. For this sample, we use the CellRanger cut. And then this is the analysis ID that we used when we were analyzing the data. And under the analysis ID, we could see some alerts. In this case, there is one informational notice to inform us that introns are included in this analysis. If your results are abnormal, you may starting to see warnings or errors in the alert section. Each web summary could have multiple tabs, depends on what library types we have included in the analysis. Here, we only have a gene expression library, so we have two tabs, Summary and Gene Expression. The Summary tab includes metrics that describe sequencing quality and various characteristics of the detected cells. The Gene Expression tab display information about the secondary analysis results, which can help us to determine the sample performance. So let's get started by looking at the metrics in the Summary tab. In the Summary tab, we're going to first look at these three key metrics that we highlight here. We chose the three metrics because based on our experience, these are the most useful metrics for us to get a really quick idea on how the experiment went. The first metric is estimated number of cells. This is the number of cells that are detected or specified in this analysis. Here, because we use force cell option when we're running analysis, therefore we have 8,000 cells as how we specified in the analysis. And the second matrix we're highlighting here is the mean reads per cell. This is calculated by using the total number of the reads in the library divided by the number of cells. We recommend a minimum of 20,000 reads per cell. Lower values here could indicate low sequencing depth. The third matrix we want to highlight is the median gene per cell. This is the median number of genes that are detected among the cells. This matrix is dependent on the cell type and also sequencing depth. Here we observed 190 genes per cell, which is lower than what we would normally expect. But we think this can be explained by two factors for this specific sample. Number one, we included neutrophils in this dataset, We know that neutrophils could express only around a few hundred genes based on literature. So it is expected that the median gene per cell is lower. Secondly, we use the force cell option here. So when we force 8,000 cells, we know some of the cells that we counted here could be background, which normally have lower number of genes per cell. So now let's move on to the sequencing metrics. In this video, we're not going to go through all the metrics individually, but only focus on some key metrics in each of the tabs. If you're unsure about what a given metrics mean or what's a plot mean, you can click this question mark to expand the definition box to see what each of the metrics mean. And here we're only going to highlight a few that are highly relevant for this specific sample. In this sequencing tab, the first matrix I'm going to highlight is the valid barcode. This matrix shows the fraction of reads with barcode that match a whitelist. The whitelist means a known list of barcodes that are included in the assay kit. 98% is pretty good. If you see a lower number here, for example, less than 75%, it may indicate sequencing, sample quality, or library preparation issues. Next matrix I want to highlight is the sequencing saturation. This is a measurement of the fraction of library complexity that was sequenced in this experiment. Depending on the goal of your experiment, you may or may not need high sequencing saturation. Here, 90% is pretty high, which is more than sufficient for us to achieve the goal of this experiment, which is to identify neutrophils and explore the neutrophil maturation. So now let's move on to the next box for the mapping matrix. The mapping matrix could vary a lot depending on the species you are working with, depending on how good your genome assembling is and how good annotation is. Overall, we would like the reads mapped confidently to genome to be pretty high, 85% or even higher for mouse and human is pretty normal. And the next matrix I want to highlight is the reads confidently mapped to intronic regions. This can be slightly higher when the sample is prepared with nuclei or the sample have high level of intron retention. Based on literature, we know neutrophils can have higher intron retention, so we believe this slightly higher number of intronic reads could be explained by that. The last matrix I want to mention in this box is the reads mapped confidently to transcriptome. So this is the matrix indicating the percentage of reads that are considered for the UMI counting, which is the final results in the analysis. And the 76% here is very good. Overall, the mapping for this sample is pretty good. So now let's move on to the next matrix box for the cells matrix. And on the top is the barcode rank plot, which shows the distribution of UMI count in each barcode. I believe this is one of the most important and informative plots in the web summary. Before we dive into this plot, I would like to show a couple examples so that we can better understand this. We have some good examples in one of our technical notes. So let's go find the technical note first. So if you go to the 10x Genomics website, and we can go to the search box on the top right to search with the term web summary. Then it will show a couple documents about how to interpret in web summary. because we have several different pipelines, so there are several different of this document. Today, because we're looking at the cell ranger, we're going to open the interpreting cell ranger web summary file for single cell gene expression assay. Let's click View and open this technical note. This technical note has lots of great detail about how to interpreting all the metrics on the web summary file. And let's jump to page number 4 to look at the barcode rank plot. I'll make this a little bigger. So on the top left, we're looking at a typical barcode rank plot. On the y-axis, we can see this is plotting the UMI counts. On the x-axis, we can see these are barcodes. The barcodes are ranked based on the UMI counts, from higher to lower. And the barcodes that are colored in blue are the ones that are identified as cells. And the barcode in gray are the background. A steep drop-off here indicates there is good separation between cell-associated barcodes and background. So in good examples, we expect this sharp cliff here. So we also call it as a cliff and knee structure, where the cliff is the transition between cell to the background. And then the knee marks the background area. So this is what a typical good barcode rank plot looks like. And there's another possible scenario is if we have a heterogeneous sample, for example, a sample with multiple different cell types that have different level of gene expression, we may see more than one cliff and knee structure. This is actually pretty similar to our example here. So you can see we have multiple cliff here in this barcode rank plot. And we think this is totally reasonable for this specific data set we're looking at. We have PBMCs in this sample, which tends to have higher gene expression level. We also have neutrophils, which have relatively lower gene expression level. And also because we set 4 cell to 8,000, we might get a little bit more into the background, but that's okay. We're going to filter them out in later steps. But overall, we could still see a nice separation between cells versus background. So for other metrics in the cells section, we have already highlighted some of them in the key metrics section. The only other metrics that I'm going to mention here is the fraction of reads in cell. This shows the fraction of valid barcode confidently mapped reads in the cell-associated barcodes. If you see a percentage lower than 70% here, it may indicate high level of ambient RNA partitioned into the cells. And here we have 90%, which is again pretty good. The last section in the summary tab is sample section. Here we include basic information about the specific sample and the input parameters that we used for this analysis. It is always useful to double check a couple things. For example, if the chemistry version detected is consistent with what you used in this experiment, and also whether the transcriptome reference path is what you are expecting. In terms of the pipeline version, we in general recommend using the latest pipeline available. So this is all I'm planning to show you in the summary tab. Let's move on to the next gene expression tab. So the gene expression tab contains information on secondary analysis. You can see it includes t-SNE projection, and also include top features by cluster. This is differential gene expression analysis results. The goal of this tab is to help us to quickly assess the quality of the results with some biological context. It is really not intended for downstream analysis. You should carry out downstream analysis using other tools such as Loop Browser, Seurat, or other community-developed tools. So let's take a look at the t-SNE plot here first. We have two t-SNE plots on the top. The one on the left is a t-SNE projection colored by the UMI counts. The brighter colors and the redder colors are the higher UMI counts, and the blue colder color represents lower UMI counts. And on the right-hand side, we have t-SNE projection colored by clustering. By default, we're showing the graph-based clustering, but you could also toggle to see the k-means clustering results. So for the t-SNE plot here, we're looking for t-SNE plots with well-defined structures. Different clusters are separated from each other is a good thing. If you see a sample with lack of structure or just a big, large cluster, it may indicate the sample quality is poor or there's loss of single-cell behavior. The next section we're looking at is the top feature by cluster. This shows the top differentially expressed genes in each cluster showing on the top right. There's so many clusters here. To keep it simple, I'm just going to use k-means equals to 4, so it's less clusters for us to look at. So this table shows the top differentially expressed gene in each of these clusters, and this again is not intended for doing downstream analysis such as cell type annotation. It's really just to help us to understand whether we have clusters of cells that are relatively poor or they make sense biologically. So in fact, the first cluster we're looking at here are this cluster in blue on the top right. And if we look at the t-SNE projection based on the UMI counts, this is also the cluster of cell with very low UMI count. And when we look at the differentially expressed gene, we can see many genes in this cluster are start by empty. These are mitochondrial genes. This may indicate some of the cells in this cluster may be unhealthy or they're dead cells, which is okay because, as I mentioned earlier, we're going to run some filtering later on. But again, this is just a quick and good way for us to understand the overall quality of our sample and may also direct us to know what are the additional steps we need to do for the downstream analysis. So this concludes what I'm planning to show you guys on how to use web summary to assess the sample quality. But before we move on to the next section, I would like to show a quick comparison of the results from here versus using the default parameter. So when we go back to our cloud analysis, we have already analyzed this data with the 4-cell custom parameter. We also analyze the data using default parameter. So I'm also going to open the web summary using the default parameter. I'm going to put these two web summary side by side. I'm going to first make them a little smaller, and then I'm going to put them side by side. So the web summary on the left-hand side is the results from default parameter, which is letting cell ranger to tell us how many cells the peptide can detect. On the right-hand side is what we've just explored, the results using 8004 cell. The first thing we can immediately see is the estimated number of cells is very different. Using default parameter, we were only able to detect less than 2,000 cells. If we look at the barcode rank plot, we can see using default parameter, we were only able to capture the cells that are in the first cliff, and we missed many of the cells here, which are likely neutrophils because neutrophils have lower gene expression level overall. And because we have less cells, some of the other key metrics also changed pretty drastically. For example, the mean reads per cell is much larger now. This is because the total number of cells is smaller. Also, the median gene per cell is much higher than before. This is because with default parameter, we were only able to capture high expression cells such as PBMCs. They in general have a higher number of genes per cell. So this is just a quick comparison of how different the results could be, and in your specific analysis, our general guidance is you should try run your analysis using the default parameter, which is letting Cell Ranger decide how many cells you have in your sample. If you're not satisfied with the results, or like this case, if you want to capture some of the cells with very low expression level, you can try the 4-cell parameter. And when you set the 4-cell, we recommend to set the number of cells you are expecting in your experiment. If you are unsure, it's better to overestimate at this stage. In this dataset, we forced 8,000. We did potentially included some background and some empty droplet, but that's okay. In the next section, we're going to use Loop Browser to filter out the background and also annotate the cell types in the data. Thank you very much for your attention.